The life of a cell can be explained through the phases of the cell cycle. As new cells are needed, existing cells divide to produce more cells. The longest stage is interphase, which accounts for roughly 90% of the cycle. Interphase can be broken up into three subphases: the G1 phase, in which the cell grows, the S phase, in which the cell continues to grow as its chromosomes are copied, and the G2 phase, in which the cell is still growing as well as preparing for cell division. After interphase is the mitotic phase, which consists of mitosis and cytokinesis. Mitosis can be separated into four stages: prophase, metaphase, anaphase, and telophase. In G2 of interphase, there are two centrosomes with centriole pairs formed by the replication of a single centrosome. The duplicated chromatin and nucleolus are bound by a nuclear envelope. During prophase, each chromosome consists of two identical sister chromatids, created during DNA replication in interphase and held together by centromeres. The mitotic spindle begins to form and the centrosomes move away from each other. In metaphase, the centrosomes are at opposite poles of the cell, and spindle fibers run from both poles and are attached to each centromere. The nuclear envelope has broken down, and the chromosomes are now lined up along the metaphase plate, which is comparable to an equator equidistant between both spindle poles. During anaphase, the centromeres have divided and the chromatids have become chromosomes. As the kinetochore microtubules shorten, the separated chromosomes begin to move towards opposite poles. During telophase, a cleavage furrow forms and chromosomes uncoil, becoming less condensed. Two daughter nuclei form and nuclear envelopes develop in the cell. Finally, during cytokinesis, the cell divides and results in two diploid cells with genetically identical nuclei to that of the parent cell. The cells have two full sets of chromosomes. In meiosis, the number of chromosome sets in the cell is reduced from diploid to haploid, meaning the cells contain only one of each type of chromosome and therefore have just one set. In meiosis 1, there are four stages. In prophase 1, replicated homologous chromosomes pair and exchange segments. Crossing over occurs, and therefore each homologous pair contains one or more chiasmata. Spindle fibers begin to form, and the nuclear envelope breaks down. During metaphase 1, chromosomes line up by homologous pairs along the metaphase plate. The chromatids from each homolog are attached to kinetochore microtubules from one pole, as those of the other are attached to opposite poles. In anaphase 1, the shortening of the spindle fibers cause the homologous chromosomes to separate and move towards opposite poles, while the sister chromatids remain attached. This cuts the chromosome number in half. In telophase 1 and cytokinesis, two haploid cells form, while each chromosome still consists of two sister chromatids. Meiosis 1 completes the separation of homologous chromosomes, which differs from meiosis 2, which then separates sister chromatids. In prophase 2, the spindle apparatus begins to form. In metaphase 2, the chromosomes position themselves on the metaphase plate as in mitosis. The centrosomes are located at opposite poles, and the kinetochores are attached to microtubules from each pole. During anaphase 2, there is a breakdown of the proteins at the centromere holding the sister chromatids together. This allows them to separate and move towards opposite poles as individual chromosomes. Finally, during telophase 2, nuclei form, chromosomes begin to unwind, and cytokinesis occurs. This results in four haploid daughter cells, each containing one set of unreplicated chromosomes. There are considerable comparisons between meiosis and mitosis. However, they play different roles in animal bodies. Mitosis is important as it is used in eukaryotes when genetically identical cells are needed, for instance during growth, when tissues have been damaged and need to be repaired, and for asexual reproduction. Meiosis produces gametes, and due to the reduced chromosome number, introduces genetic variability among the gametes. There are other differences, such as the occurrence of crossing over in meiosis during prophase 1, while crossing over doesn't occur at all during mitosis. Mitosis has only one division, while meiosis has two, and both result in a different number of cells, as well as the number of chromosomes in those cells.